Hi, today's topic is mechanism of concentration of the filtrate. Okay, it means how the filtrate is becoming more concentric compared to the from the dilution. So first point is that in this topic we are discussing about the mammals. Mammals has a special ability to produce concentrated urine. This is the topic actually. How? Let's see. First of all, the topic starts from the Henle's loop and vasa recta. Okay, vasa recta and Henle's loop. These are two important. Okay, in the nephron, these two will exchange the materials each other. Okay. Next, what is saying? The flow of filtrate. First thing, the flow of waste in the two limbs of the Henle's loop is in opposite direction. Okay, and that is called as counter current mechanism. I'll explain. And what he's saying means here, if you see, he's saying Henle's loop. These two things are. This is Henle's loop. Okay, this is Henle's loop. This one. This is Henle's loop. It's going this direction. Okay. So this is a descending link, Henle's loop, and this is a upside ascending link. These are both belong to Henle's loop. So that's what he's saying. Both will be very near, and this will be opposite directions. Okay, two limbs. One is down, one is up. In the case of the Henle's loop. Same way, in the case of vasa recta also two limbs. This is vasa recta. This is a blood capillary. It is also going in the two directions. Okay. Remove this. What about this? In this case, it is going like the blood will be moving from the arteriole, afferent arteriole, from this direction to this, then this to this. So this is movement of the blood. In the case of movement of the filtrate will be in this direction. So what is explaining that even this Henle's loop also will show counter current mechanism, and the vasa recta also will show counter current mechanism. So this Henle's loop is showing counter current mechanism. Current means here flow. So it's a descending and ascending. The same way vasa recta also shows counter current mechanism. This is the topic here in explain. One thing in Henle's loop, what will go? Okay, if you make a line, in this filtrate will go. In this vasa recta, what will move? That is blood will move. There is only difference. Okay. Now make notes. What the notes will be here. Third. Okay. That is mechanism of concentration of filtrate. Okay, how this happen? First point is mammals. Mammals has the ability to produce concentration of concentrated, or you can say concentrated urine. This is the first point. Now second point how to write, that is Henle's loop. First of all we should discuss Henle's loop. Okay. What's the Henle's loop? It has a one descending limb and one is ascending. So because of these two, it is showing counter current mechanism. The same way, third is what? That is vasa recta. It is also showing ascending limb. First, we should start from that side, ascending and descending. So this is also showing counter current mechanism. Okay. So what's happening here in Henle's loop? What is moving? It is moving, so blood. Okay, Henley's loop is actually filtrate, vasecta blood, so filterate. Filtrate is moving from descending to ascending to further. Is a movement of the filtrate. The same way here, in the case of vasecta, the blood is coming. Okay, that is blood is coming from the descending to the ascending. That is blood flow. Blood flow. So this is, you have to understand, that is just visa, Henry's loop, descending, blood is coming from down to going upside. Okay, so between these two also counter current mechanism. The vasecta from the upside, that is a descending loop to the ascending loop. This also counter current mechanism. These two we explained here. Okay, that is vasecta is also showing in a counter current pattern. This is the point. These two. So you should understand the image also like this. Based on this image, you should understand this notes. Okay, this is Henley's loop, descending and ascending. Vasecta, descending and ascending. Now we'll see next topic. That is the proximity, number one. And between Henley's loop and vasecta, I mean they are very near, as well as a counter current mechanism in the help in the maintaining and increasing the osmolality. It is making the osmolality in the inner medullary interstitium. So what's helping? Number one, that is a proximity. Second, counter current mechanism. Because of these two, what's happening? That is, there is osmolarity is increasing. This is answer. Okay. So the topic starts here. Fourth point. That is, what is making increasing osmolarity? Okay. Osmolarity increases in inner medulla. Okay, how? First question. How? Answer is between two. One is the proximity. The proximity very near. They are very near. You see, they are very here. There same thing. This is the Henley's loop. It's down and going upside. Here, vasecta. It's down, going upside. This will be above it. Okay, like totally wrapping. So that's what. That is counter current mechanism. So while wrapping, it will be one is proximity. Second is its counter current mechanism between two. Counter current mechanism between Henley's plus vasecta. This is the next point. Here is explained. Next. After that, this topic completes here. So because of this, what's happening? The amount of the osmolarity will be, next is less in the cortex. Okay, and more in the medulla, in the middle. 300 in the outside, 1200 in the down. See, this is the same image he explains in this. That is in the cortex region. This is cortex, this is outer medulla, inner medulla. See, in the cortex 300, outer medulla 600, inner medulla 900 to 1200. It is decreasing. Okay, why? Because of the exchange. See, here this is the ascending limb of this filtrate. It is releasing NaCl. Okay, and that NaCl is taken to the this descending vasa recta. It's going taking back, again taking upside, again it is going into the medulla. So because of this in the medulla, the concentration is more, that's 1200. It's rotating continuously within this itself. Okay, that we'll see. Not only that, even the urea is releasing from here, it is entering back into this same here, and it is taken to the collecting duct, and it is released back to the medulla. So these two things we'll see. Okay, now the gradient mainly caused by, so how this gradient occurring? Because of the two things. One is, so this difference, okay, one is NaCl, the action between the NaCl and urea. So here topic is here, NaCl and urea. The gradient is mainly caused by should note down it. Okay, let's make a point. Fifth point. Fifth point. Reason of osmolality. Okay, I think it's a gradient difference. One is NaCl. Okay, of course, this is in the inside medulla only. Okay, this is the outside is the also a reasons. Okay, this is the second reason. NaCl. What's happening in NaCl? F2. Okay, this NaCl is moving from ascending. Okay, Henley's. Okay, we'll discuss. First, what NaCl is transported? How? In the case, NaCl is transported by ascending limb of Henley's loop. Means while going into the collecting duct, okay, it's transported from Henley's loop ascending to the descending limb of vasa recta. Okay, again the NaCl is returned to the medulla. Okay, by ascending portion of the vasa recta. So you remember, better I'll write here. Okay, ascending from this part, NaCl is going from ascending Henley's loop to the. Okay, next is to the descending. I should better change the color. Okay, and we'll take a contrast to the descending of vasa recta. Then again it is going back to the ascending of vasa recta. Okay. Okay. From there it is going to the medulla. See, the NaCl is transported ascending limb of Henley's loop. This ascending Henley's loop to the where descending limb of vasa recta. This is descending vasa recta. Then again it's going to the ascending portion of vasa recta. Then from there it's going to the medulla. This thing you have to understand. I'll show here. You read the lines again. What is showing? Saying that this NaCl, this is the so this is what as a descending limb and ascending limb of Henley. This is Henley. From this ascending limb, NaCl is going, and this is very near. So this NaCl is going to the descending limb of the vasa recta. Okay, I'll explain. This NaCl is going towards the vasa. Not only this, high amount. See. Next, this is moving in this tissue direction. It's the descending limb of what? It's a vasa recta. It's going down, and see here, it is releasing. Not much. Okay, here it's releasing. It is what? It's a medulla now. This is what? It's a medulla. It's everything. Inner medulla. Even it is going upside, not in cortex, here it's releasing. Okay, so what's happening? It is not going inside. Okay, inside this one. So in the medulla it is releasing. So this is a cycle. From ascending Henle, it's going to descending vas recta, descending vas recta to the ascending vas recta, or ascending vas recta to the again to the medulla. So this is what he's saying. So this is because of this movement continuously. Here they will maintain the gradient, and because of that gradient, it will be more 1200, 900, 1200, 900. The first point. Okay, that's what we discussed. NSL is going from ascending limb of Henle's loop to the descending limb vas recta nearby. Then it is going to ascending portion, then returning to the interstitium, means medulla. So this is the NSL role completed. Next, another role. That is what? Vas recta. That is completed. Next is urea. Second is urea. What is the story of the urea? Let's see. Here he is saying the topic is like similarly saying it's a small amount of urea. This urea enter thin segment of the ascending limb. In the ascending limb again thin will be there, thick will be there. It means thin. In the where actually limb of Henle's loop, that is ascending limb. This you should remember. So urea is entering to the, means from where is entering also you have to understand that. I'll say entering into Henle's loop. From where to enter? Generally from medulla only. From medulla it's entering into ascending limb, then which is transported back to the medulla from the collecting tube to the interstitium. Okay, so number 1, to make a numbering, it will be number 1 interstitium to the ascending limb, from ascending limb to the collecting tube, again to the interstitium only. So this is cycling. Okay, I'll explain. See here what's happening. I'll take a color. Let us take a this is urea. It is entering from the, this is collecting duct. From this it is entering to here. From there it is entering Henle's, not somewhere else. This is not the vessel. The blood it will not enter. This is the blood. Okay, it's going actually it's blood vessel. Vasa recta will contain blood. Okay, it is filtrate. Entering into the thin. This is thin chamber, not here thick. It's entering into this ascending limb of the Henle's. Then it's going back like this. Okay, it's coming back like this. So this is another cycle. This is a urea movement cycle. So same thing you explain here. Hope you understood. Now let's see. Ascending limb of, okay, small amount of urea is entering thin segment. From where to entering means it is the medulla only. Okay, it's from medulla. From here this medulla. From there to the here to the collecting duct, again back to here. So that is the line. So let's write. From where is entering to, that is from medulla to where ascending, okay, ascending Henle. From there it's going to be collecting duct. This is DCT actually. Okay. Okay, next it is the DCT. Next it will be, we can say, of course, what is there? We can say DCT. Then it's going to the, without DCT cannot go to collecting duct. Collecting duct. From there it is going to the back to the medulla. Okay, Interstitial. These are urea method. So hope you understood. These are notes for this. Next, what's the main use? Okay, the above described transport of substances facilitated by the special arrangement of endless loop and vas recta. Arrangement of endless loop and vas recta. Even these two also in the counter current mechanism. Not only their limbs, okay, even these two, because that it's happening easily. Next, this mechanism helps maintain a concentration gradient. Okay. In the medullary, okay, because of this continuous movement and settling in the medulla, gradient will be more. Gradient means it's a difference, high amount or low amount. The presence of such interstitial gradient helps in the use of this gradient means difference of this interstitial. What is helping? Okay, one, easy passage of water. This is a use. Next is concentrating filtrate urine. Second use. And because of this, the urine is produced 4 times concentrated than the initial, that's PCT. So these are 3 uses. So what is the topic? This interstitial gradient is helping. How? That is the answer. Okay, let's make a notes. 6th point. Gradient. Okay. Difference in medulla. Especially, okay, interstitial means very inside. Helps what? One, easy passage of water, that is actually along the urine. Okay. Second is concentrating of filtrate. That's nothing but using. Third is 4 times concentration of urine. Okay. And initial PCT. These are notes. Okay. So in this topic explaining mechanism of the concentration filtrate. One is mammals have concentrated urine. Why? Because the arrangement of the Henle's loop and vas recta. Again, these are two will move both quite opposite. Okay, one will move the waste, one will move the blood. Next thing is they are very near proximity. Their closeness is very near. Okay, that is very near in presence. Second is movement. So if it's something it is while ascending, it can have to release, they'll release into descending limb. Again, this ascending limb again it will release into descending limb. So it is also a cycle. This cycle helps. That is this counter current mechanism helping. So while helping, so always because of gravity also, okay, because of this between this organ arrangement also, always this NaCl and urea will be going to the center of the medulla, that is interstitial. Okay. So how the NaCl is moving from the Henle's loop to the blood vessel, that is vas recta, again to the medulla. So it is extra means again this will be taken, sometimes sodium chloride will be taken, absorbed or released through the from this PCT also. So that also should remember. Again urea from the medulla to the ascend Henle loop ascending to the DCT collecting duct, again medulla. So this is the two flow of the reaction. Our reactions are the movement of the materials, sodium chloride and urea movement. Next usage. Hope you understood the notes. Thank you.